So I'm from the other London in Canada. It's got a River Thames, it's got an Oxford Street, it's got a Covent Garden Market, it's got a Pall Mall, it's got King's College, and it's only got half a million people. But it has, well, just for those of you who are geographically challenged, it's just to the left of New York and to the right of Michigan, about 200 kilometers away from Toronto, over here, Detroit and Cleveland, so equidistance from those three cities. Western University, where the Robarts Research Institute is situated, has about 36,000 students, occupies a very large 4.5 square kilometer plot of land on the River Thames. This is what it looks like in summer, this is what it looks like now. It was minus 23 there yesterday, but it was plus 36 in the summer, so we have a slight temperature range. It has medical engineering faculties, very strong medical engineering faculties, as well as law and business. Very fortunate to have a hospital, a very large general hospital on site, connected physically to the medical school and the Robarts Research Institute. The institute itself has about 600 people, roughly split between 50% imaging and 50% molecular medicine. The imaging portfolio pretty much reflects the sorts of things that you do here at Imperial, they do at UCL, they do at King's. But we're all in this relatively small community. We have, just in the research building, these are not clinical machines, at 9.4 T 20 centimeter bore animal MRI, 7T human MRI, 3T human MRIs, an NHP, non-human primate colony. So we do a lot of research with marmosets, macaques, and chimps. Also within the institute, with interventional radiology and interventional and surgical suites, research suites, and very tightly integrated with the Western School of Biomedical Engineering, the Department of Medical Biophysics, and the Department of Medical Imaging, otherwise known as radiology. So a very active, comprehensive imaging research group at Robarts. My lab, the VAST lab, Virtual Augmentation and Simulation for Surgery and Therapy, that was a cool acronym that one of my staff came up with about almost 20 years ago when it kind of starts, so I have to write it out because I never remember what it stands for. But we do a lot of image-guided interventional work. It started off just being focused on the brain, but it rapidly expanded to heart and other organs as well. And so, for this talk, I'm going to talk about how we got into the whole patient-specific valve area through some of the research that we've been doing over the last few years on minimally invasive image-guided cardiac interventions, and then talk more specifically about the cardiac simulator itself. And I think these images kind of portray why we need to do better when we're performing minimally invasive interventions on the heart. This first, let me just run that one again. Maybe not. You probably saw the stent being navigated towards its final resting place, and you might have seen a very quick puff of contrast to help define the aortic outflow tract. But otherwise you don't see anything. You don't see the heart, you don't see the aorta, and you're driving blindly. Alternatively, you might be using transesophageal ultrasound down the esophagus, looking at the heart from behind, and looking at the manipulation of some instrument inside this happens to be inside the mitral valve annulus. But you're using that image, or maybe a pair of orthogonal images, to try to navigate an instrument into that critical space within the mitral valve. Neither of these are particularly intuitive. And when things go wrong, you can see again the valve is being expanded by a balloon. when the cardiologist thinks it's in the right place, he deflates the balloon, and ideally that valve will stay put. But as you see here, the minute he deflates the balloon, that valve starts to move. And a few seconds later, there it is bouncing around in the left ventricle, both in X-rays and an ultrasound. Not a good outcome for the patient. I mean, fortunately, they had to go in and do an open heart procedure and extract the valve, and then do the procedure again in a completely different way. So the patient was fine in this case, but it took an awful lot of extra effort to save the patient. So this was the motivation for the work that we'll be doing over the last few years on cardiac interventions. And it can be boiled down to the surgeon being completely disoriented. He's looking at images that are completely disconnected from his proprioceptive field. He's moving instruments, either whether it's a catheter or a rigid instrument, through a slot between the ribs, through the heart wall, and he's trying to figure out what's happening to that instrument by looking at these images, either the X-ray images or the ultrasound images. So he kind of knows what he has to do, but he has no idea what he has to do to get that desired effect. And so in the meantime, you're poking this probe around inside the heart, hoping eventually you'll see it and be able to place it in the right place. So our cardiac surgeons came to us. They needed help on these problems. And they were sure that what they needed was the dynamic preoperative cardiac MR image displayed in 3D, fused with ultrasound, fused with the tools, and that would solve all their problems. So that was a great PhD, a bunch of PhD projects, and we did a lot of work on that. So we ended up, we tried to look at what we could do to make improvements to these procedures by augmenting ultrasound with other graphics components that were representative of targets within the heart. Throughout this talk, I'm going to use AR, or augmented reality, in a fairly loose form, because in this context, I sort of consider the ultrasound or the X-ray images as being reality, and then we augment them with something else. So it's a sort of different dimension to augmenting a normal physical reality with some other image. But we use the term AR anyway. So could we, in fact, use this sort of augmented reality technique to reduce operating time, increase patient safety, and even eliminate the need for X-radiation and the X-ray contrast that are normally required in cardiac interventions? So the first manifestation of this was this kind of display, which should be 3D beating heart, segmented chambers from MRI, fused ultrasound, and the reaction was, I have no idea what I'm looking at. There's far too much information. So lesson one is iterate from the start on what the surgeon needs, not what he thinks he wants. Because he'll always give you something that's either impractical or to build, or impractical from the sense of him interpreting the images. So that taught us a valuable lesson, and we went back and made sure that we understood what the surgeon needed at all parts of the procedure. And it also told us that there are for many minimally invasive intervention procedures, there are kind of two distinct pathways. First of all, you've got to get to where you need to be safely. And then you've got to perform what you need to perform. So it's like using a GPS to get from my home to a parking lot. And then once you're in the parking lot, you've got to get into a very tight space. So two different imaging or tracking modalities are often required. And once we understood this, it meant that we could divide the image-guided surgery problem into a navigation and a positioning phase. And that kind of changed the way we looked at things. So a particular procedure that's been around for a while that uses a rigid instrument to sew new cords on a flailing mitral valve leaflet. The leaflet is flailing because the cords that normally normally restrain the excursion of the valve, they stretch or they break, and instead of doing this, it does that. And so instead of constraining the blood and pushing it out the aorta, it gets pushed back into the left atrium. So you'll see a little bit more about this as we go on, but this technique is currently guided by a pair of images, orthogonal images, that come out of this transesophageal probe, and they're displayed on the screen like this. And so this is exactly the situation that I showed before, where the surgeon is moving that stick around inside the heart, trying to see where he's going. So here's the stalk going up, and what the surgeon's trying to do is to get this in the region where he needs to be to actuate a little mechanism at the tip of that stalk, which grasps the valve and sews a little thread through it. It's like a sort of miniature sewing machine up in the top. And if you get that to work, it works quite nicely. You pull that thread back and you tension it appropriately and you tether it at the apex, and the valve starts working again. So this is what he's trying to look at on the way to getting to the right place. And this is, in this particular image, he's in the right place. But if you track the tip of that instrument, and this is over 6 different surgeons performing this procedure using the standard of care in a pig model, pretty much you see an outline of the left ventricle. Eventually they get it right after lots of retries. Sometimes they end up going through this red annulus. Anyone suggest what that is? That's the aortic valve annulus. That's where you don't want to go. That's why it's red. You want to go through to the green annulus, which is the mitral valve annulus. I should mention that these rings are defined on the ultrasound images prior to the procedure. We have a little simple app that allows you just to use those 2D planes and move them around and click on a number of landmarks around the annulus of each of the mitral and aortic valves so that we get those registered in 3D space. So that's the result. So we went back and spent a lot of time talking with surgeons to understand exactly what they needed to in a platform that would help them navigate successfully. So we started off by magnetically tracking the transesophageal probe and the instrument. So we have a magnetic tracking transmitting system on the patient's bed, little 5 or 6 DOF magnetic tracking transducers in the ultrasound probe and in the instrument. As I mentioned, we identify the target annuli. We add the position of the tool and the ultrasound to the biplane images, now reformatted in 3D, so you understand the context between what you see in one image and what you see in the other image. We added an axis to the tool so that you knew that if you lined this axis up and it was pointing in the right direction, you just had to follow that line and you'd end up in the right place. We added this. We added the green and red to identify which is the target, which is dangerous to go. We gave these 2 views, the bullseye view and the side view, to see how close to being on target you are and how far away you are from the target. And so we set this up, and now using this platform to perform the same procedure, and we had 6 cardiac surgeons performing these procedures, one of whom was the surgeon from Mayo Clinic who had actually developed this device originally in the first place. And with this platform, instead of the ultrasound, we ended up with combined tracks looking like this. Otherwise, they went pretty much straight in. Now, I do point out that that in the platform, these annually are stationary. We did have a version of the program that had them move with the heartbeat, but these procedures are performed in a fast-pacing situation in any case, and so there's very little movement. And you're not trying to accurately target any point in here. You're just trying to get to the center of that annulus, because once you're there, then you can switch imaging modalities to refine what you want to do. So here's the protocol that we used for the surgeons, tracking the torque paths every half a second. And this is what it looks like in the OR. You see the raw ultrasound here, you see the platform up here, and the surgeon is looking directly at this platform while manipulating the instrument inside the pig's chest. When we analyzed the data, we were quite amazed at how much difference this very simple change in imaging platform made. So that the distance of errors from the midline reduced by a factor of 3, navigation time by a factor of 4, and the total path length by a factor of 5. And so this manifested itself in, let me just say one thing first. This is a single trajectory from the inventor of the original device, who's been using the biplane ultrasound forever to perform the procedure. This is the trajectory when he's using the standard of care. In one case, he actually went through the left ventricular outflow tract. In other words, poked the instrument through the aorta instead of the mitral valve annulus. When he turned the new platform on, he just went straight in. When we looked at the times that the tip went to parts of the heart which were potentially dangerous, we found that there was a 40 times improvement in patient safety. Well, let me just stop at that point and give you a bit of an anecdote, a follow-up anecdote. So you'll be surprised to hear me say that the standard of care 5 years later is still 2 ultrasound images. Why? Well, the neocode company that we were working very closely with, we had license agreements, patents, a lot of funding, eventually fired their CEO and CTO, the guys who were interacting most closely with us and who were very, very excited about this augmented reality platform. And they completely dropped the relationship with us and went forward just with the way they were going. Admittedly, there was extra cost involved in implementing our solution. They had to put magnetic tracking sensors in both the ultrasound transducer and the tool. And it also meant that they had to go through a whole new set of FDA and European regulatory processes to get the new system funded. So it was, to a large degree, I think, was also financial. They were not a very big company. So it's a cautionary tale. Even though you feel that you've got a winner and you've got a company that's fully backing you, it can sometimes all fall apart. Anyway, having gotten to that target point, we can now change our imaging modality and use something different to do the positioning. And in this case, we placed a very standard intracardiac echo probe inside that instrument so that we could look out through the jaws, and we could see the flapping leaflet and really get a very clear idea of where that instrument, where that tool is grasping the valve, just before we poked our needle up through the center of this device, grab the cord that's sitting in the top here and pull it back down, forming the knot. And using this, we can even can help us identify where we can place multiple cords, because not only is it one cord that we use, we often use more than one cord to perform this procedure. So you can drop an object in 3D space at the position of the first cord you've made, and then move the tool around by so many degrees to identify where you need to put the next one. So that was the technology for mitral valve implantation. We also looked at how valuable this type of approach would be for implanting aortic valves. There are a number of different ways of putting aortic valves into the aortic annulus. There's different types of stents that can go in, and these are delivered either via the apex or via the vascular system, entering from various vessels in the body, from the aorta from the carotid directly, for example, from the arteries in the leg. And we demonstrated that we could use the same sort of technology, again, identifying those annuli with ultrasound, and using our track system to go directly to the target. And because we know where the target is, we know where the instrument is, we can change the color of the target as soon as we get to the right place. So it's going from red to green. We can expand the valve and be assured it's in the right place. So this was demonstrating it for apical access. We can also do the same thing for, sorry, these images didn't come out very well. Through the vascular access for the transfemoral procedure. And it's the same sort of thing happens, and so the green disc turns red. These look terrible, sorry for that. Anyway, we did a lab bench test at least to evaluate whether this type of approach using ultrasound alone without any x-rays and without any contrast agent could do just as well as the standard of care that does use x-rays and contrast agent. And in a relatively simple study, we nevertheless determined that we could do just as well with ultrasound as we could with x-rays. So this opens up the promise for a new way of performing this extremely frequent procedure of implanting a new aortic valve, just using ultrasound. And we're trying to work with companies to get some buy-in to do this. And again, we noticed that because companies have a vested interest in selling the things that they've been selling for a long time, X-ray companies have a vested interest in X-ray-based catheter labs, it's difficult to get companies in this business excited about these quite new ways of doing things. So, how does this lead up to heart valve models? Well, the basic point is that pigs are pigs. And a lot of this development and evaluation was done on porcine models in an extremely well-equipped animal lab in the hospital just adjacent to our institute. But it's hugely expensive. A lot of people have to be involved. A lot of regulations, a lot of transport issues, disposal issues, and so on. And even when a pig is on the operating table, maybe you're poking things into his heart. They're not necessarily particularly cooperative. Sometimes they'll go into atrial fibrillation in the middle of the procedure, and then you're done. Sometimes they just die. So, in view of this, we developed our internal solution, and this was originally developed to accommodate mitral and aortic valves. This is the left ventricle pumping, and in the flange just up inside here, we have we can put either purely artificial valves or patient-specific valves, either atrial, mitral or aortic, and use this instead of the pig for evaluating our procedures. In fact, since we built this first prototype 5 or 6 years ago, we haven't done a single pig study. But this whole project kind of took on a life of its own when we realized that, or when not only us, but other people around us realized that this could be used for evaluating the efficacy of performing a procedure on a patient-specific valve. So we built, this was the original simulator, we've refined it. Most of the time we're not performing procedures through the apex, but we're just interested in how well a valve performs under realistic beating heart and pressure gradient conditions. And so this model has been somewhat superseded by this model, where we have a motor pushing a flange in and out to simulate blood flow in this sort of pressure chamber to help us regulate the different pressures within the cardiac chamber. So back to mitral valve surgery, this is continues to be a problem because of prolapse or failure of the valve within quite a significant percentage of the population. So the current clinical workflow is to look at the Doppler transesophageal echo image, also perhaps look at the cardiac CT image, and make some sort of judgment about replacing or repairing it. And when you've made that decision, do we do it on a non-pump situation or a beating heart situation? On-pump means the patient's heart is arrested, the patient's heart and lungs have been taken over by the heart-lung machine. Not a particularly desirable thing to have happen to you anyway, because first of all, the blood doesn't like having itself pumped through a mechanical device. And you can often do damage to the patient just connecting that heart-lung machine up to the patient. So you can dislodge plaques, for example, that can either end up in the cardiac arteries or the cerebral arteries, causing a heart attack or a stroke. But these are the different options. And there are many options for repair. We can on open heart, we can just suture this ring into the mitral valve annulus to make it smaller, so that if that annulus has gotten stretched, then those valves are not going to coact anymore. But if you make that smaller again, then maybe those valves can work properly. You can put a completely new valve, either mechanical, one made of from bovine valve tissue. You can literally repair with sutures this cord. Or on the off-pump situation, you can do the near-cord procedure that I just described before. You can do this really bizarre procedure, which involves clamping the center of those leaflets. And surprisingly enough, I mean, this is also replicated in this situation up here, where you've sewn the two leaflets together. This, surprisingly enough, actually helps the valves work better. And this has become a fairly popular, so-called MitraClip, fairly popular minimally invasive procedure for repairing heart valves. Or you can actually put a stent-like valve in. It's a bit more complicated than the one for the aortic valve, but this does exist. So the difference between off-pump and non-pump, you're either coming through the vascular system, putting this clip on, or sewing this thread around the cords, or you're opening your heart up completely and have a hands-on approach to perform the procedure. So the basic question is, how do you decide on the optimal treatment? Well, we started looking at whether we could make a patient-specific valve based on the patient's transesophageal echo image before surgery, and then perform a variety of procedures on it and test it in our beating heart simulator to see which procedure was more appropriate for that particular patient. So the process is starting with data collection. This is a 3D echo image from a typical patient. Then we take that image and we make a 3D model of it. We refine the model to make sure that there are no gaps in it and it looks representative of the actual, what we expect an actual valve to look like. Then we 3D print that model. And this is the 3D printed model you see here. It's a rigidly printed model. But then we use that model as a template for literally painting silicone. And so you can see that the valve here, which had a very thin layer of silicone with some very light gauze mesh embedded to ensure that these cords could be embedded in it successfully. And then we surround it by this silicone flange. And the net result is we have the valve with all its cords embedded in the silicone flange. And then that can be placed in our heart valve simulator or beating heart simulator. So here it is in the simulator. Here's the beating valve seen visually in the simulator. And this is the Doppler ultrasound, both in the model and in the patient. Then we can give that valve, the patient-specific valve, to the surgeon. He can perform one or more procedures on a single model or multiple models of a single valve, and then place it in the simulator to see which one is going to give him the optimum effect. Up until now, TE imaging has been used as a primary imaging modality for both diagnosing and guiding these procedures. But it's not optimal for the sorts of things that we need to do, being able to get volumetric data to help you size that stent you might want to put in, or the ring. And it can be somewhat unreliable to segment the valve for patient-specific modeling. So, in other words, you get an image like this in the ultrasound, which segmentation is correct. So we looked at how well, we wanted to find out how well the segmentation that we were doing was replicating the model of the valve. We also wanted to see how well dynamic CT would allow us to image that valve as an alternative. But mainly we wanted to see whether high-speed, high-resolution CT could actually give us an idea of how well our ultrasound modeling was doing compared to the CT of the actual valve. So there are lots of challenges with imaging valves in the CT scanner. It's large and rapid motion, particularly high heart rates. We have a great deal of difficulty getting the temporal resolution correct. And is there sufficient contrast in the valve that we can do all this without contrastation? Of course, with a CT scanner, there's X-rays as well. But we can provide high-quality volume rendering to get volumetric data from the CT. So we built a rig to answer these two questions. Does dynamic CT reliably capture the morphology and behavior? And can we use it to validate our segmentation? And so here's the system that we developed. Here's the flange, here's the valve, and we had these little motors which would allow us to open and close that valve at specific parts of the cardiac cycle. So we wanted to be able to image this valve at various stages of its opening and closing, but without motion artifacts. So we had these little motors that could tension the valves to the appropriate, via the cords, to the appropriate levels during the cardiac cycle. we could also run it in full dynamic mode in the heart simulator. We also did the same thing with a bovine valve, because we wanted to see how well our silicone valve was emulating reality. So we thought putting animal tissue in there would help us answer that question. So we started off with at the degree of opening and closing of the valve at various points in the cardiac cycle. These are the static positions of the valve at these different places. And we made a number of static images. We then set the scanner in dynamic mode and captured the dynamic images at the same points in the cardiac cycle. And then by taking these images from the CT volumes, both in the static case and the phase-matched dynamic case, we could go through this workflow and look at the distance between the static images and the dynamic images. This just shows one example in the map showing the differences between the two values. We seem to have a bit of an offset here, which probably is as a result of a slight original registration mismatch. But this allowed us to see whether the dynamic scan was sufficient to describe the valve. In terms of which segmentation is correct, we looked at the bovine valve, silicon valve, wide images, both of TE and CT, and registered them, did the same sort of analysis, and compared the two. The net result was that, in general, ultrasound overestimates the leaflet thickness, particularly when we're using a silicon valve. When we have the bovine valve, similar to the patient's tissue, we get images from ultrasound that are closer to the CT than the silicon valves with the CT. And the most important conclusion is that pretty much all we really need is the center lines of the valves to accurately represent what's going on. And so, in a sense, that's what we're using when we're painting the silicon onto that solid template. We're basically extending the center line of the valve to a realistic human valve thickness. So once we've done that, we can place the valve with whatever pathology is demonstrated by the ultrasound in this jig, which allows the surgeon to perform a particular procedure on it. In this case, he's putting a ring around the mitral valve annulus to basically bring that annulus, make it smaller, give those valve leaflets a greater chance of coapting. The anatomical features that are necessary to measure, to decide on which procedure you're going to use, the size and shape of the annulus, and the amount of regurgitation that's coming out through that gap between the non-coapting valves. And on the right, you see the full procedure that the surgeon's performing. So once we've made that particular repair to the valve, we can place it into our cardiac simulator, turn on the beating mechanism, and look at the behavior of that valve over the cardiac cycle. And here's an example of what that looks like. Side view of the valve, opening and closing. This is the differential pressure between the left atrium and the left ventricle. And you can see it gets up to about 110 millimeters of mercury when it's working properly. So this is the increase. And by changing the tension on those papillary muscles, on those chordae tendineae, you can see how we can adjust the amount of closure. So down here we've tensioned it too much, and we're not getting any pressure differential. So we can optimize the pressure differential by changing the tension in these chords. So down here you see the two-dimensional and the three-dimensional ultrasound at the same time. And here's an example of these videos are going to be jerky, but showing the same valve, multiple versions of the same valve, but different repair strategies. One is the Alfieri repair, and one is the ring. And so we can see visually how these look in our simulator, opening and closing. And we can see the Doppler ultrasound in terms of how much regurgitation you're getting. So that started off as being a retrospective study. The surgeons reperformed procedures that had been performed earlier, and then tried to match what we saw with the modelled repair compared to the repair that had actually been done surgically. And we showed that there were really significant differences between pre and post repair that indicated that this might be really a valuable tool to use going forward. Again, just looking at the size of the jet, pre and post, huge differences in what we see before the surgery and after the surgery, both in the real case and in the model case. So this was a very small retrospective study which had a number of limitations, but we've published a number of articles on this. But going forward, we've got funding to do quite a large prospective study, and recruiting I think up to 30 patients that will have all of their valves modelled prior to surgery. The surgeons will actually be able to practice the procedures prior to actually performing it in surgery, and we hope to show that this approach really makes a significant difference to patient outcome. So that was just one theme in the lab. We're doing a lot of augmented reality for cardiac and other procedures, and this is one example of doing an augmented reality or virtual reality view of a beating ultrasound heart with the Doppler signal at the showing where the maximum flow is even after coaptation. In other words, the valve is faulty. We've done some work with using the HoloLens to guide procedures which involve inserting probes into the beating heart. And you can see that even with the HoloLens and all the registration and positioning the object appropriately, it certainly helps to put some black surface on object on the surface to make the human visual system believe that it's actually seeing inside the chest wall. And finally, we've been working with King's on a 3D heart project, and this is a group from Julia's group at King's, and this is involving examining a 3D beating heart model, but in this case we've got two viewers, one on either side of the Atlantic, looking at the same 3D heart model that's sort of floating in front of them. So they're both people in my lab. This is the image on the screen on the left is showing students in my lab in London, Canada, operating with that 3D heart, and we move around to the version of the heart on the screen in Julia's lab. So this is really exciting to get these two groups working on a similar project and being able to collaborate in this way. So, the basic message here, I think, is that physical models can assist in development of new procedures. And it's been this beating heart theme has been a core technology in the lab for the last 10 years almost. We can develop patient-specific valves from ultrasound. We can use them to model surgical procedures. And we can use the simulator to validate the repair. And a lot of the work that we're doing is also showing that VR and augmented reality has a potential role to play in both training surgeons and performing procedures. And that's certainly a lot of fun. Just want to pay homage to the great group that works doing these and other projects in the lab. Before I sign off, just a bit of advertising. This book came out last month. It's talking about a whole bunch of different applications of augmented reality in medicine. You can go to the CRC website to find that. And of course, remind you about Nikai. Submissions are already open. Make sure you get in before the deadlines. They're not going to change.
Thank you very much. So we have a bit of time for questions. Perhaps I can ask you, how many Mika papers have you already got in your, which have been submitted 2 months or 3 months in advance?
0. Yeah, I was just wondering if it would make what your view is on the tracking problem from your first example, the ultrasound augmentation. So say I can get rid of the tracker and solve the tracking problem with an image-based approach, image analysis approach. Would it be worth revisiting such
Absolutely.
a project?
And we're very keen to move in that direction ourselves. It would take a huge burden away from having to instrument these tools. In fact, we've done quite a lot of work on that exact problem with endoscopes. And so we no longer have to track an endoscope when we want to, let's say we have an ultrasound probe and we want to move the ultrasound probe around. That endoscope can very accurately track the ultrasound probe as well as an NDI Aurora system could, and allow us to reconstruct the 3D image in real time. So yeah, we'd love to be able to use ultrasound signatures to do the same thing, and we have great hopes that deep learning is the clue to that.
Yes.
Thank you very much, Professor. Basically, it's quite a multidisciplinary research, and I cannot even estimate the things that you showed, how long they took to actually do. What I want to ask is, in all this, what is your feeling about where is the main source of error currently? So basically, where can there be the most improvement in the imaging, in the segmentation, in the hardware, in the modeling of the hardware? Where do you feel basically there can be the most improvements?
Well, if I look at that example I gave, I think tracking the instrument relative to the target to get to the target point is probably the most important thing that we can do. And it doesn't just apply to this problem; it applies to many problems. And I think the mistake that we often make is that we provide a global 3D model of the target, and we expect that to represent the organ and the target throughout the entire procedure. We spend a lot of effort with putting biomechanical parameters into mechanical models and expecting that those are going to be right for this individual patient. So I think the first thing is to recognize that one of the most important things is to get the instrument close to where you need to be to perform that intervention. And then focus on better imaging. And by imaging, I mean not just optical imaging, we can be electrophysiological imaging, it can be spectral imaging, but using a bunch of different technologies that we have available to us to refine that target point in the end. That's where I think the field is going, should be going.
If I may rephrase, if I understand correctly, not really perhaps the method that is used to detect the model of the images, but probably the images themselves. So the image modalities that are being used and how they are used, not really whether you use deep learning or random forest to actually find it, but whether the images or the strategy for using them to register them, let's say, to the patient model, whether it's correct or not. That's what I understand.
I mean, it can always improve, and the amount of improvement you need depends on what you're using that registered image for. If you're just using it for context during the navigation phase, then it's 5 millimeters is probably fine. If you're using it for targeting, for positioning something exactly on target, then 1 millimeter may not be enough. And so, particularly in the brain, and again, another group at King's is working very aggressively on trying to predict the location of electrodes on a deeply implanted electrode within the brain, taking account of bending that might occur, for example, when you're placing the electrode in.
Mm-hmm.
So, yeah, it depends on the context. But we have to learn not to overthink these problems as well, because sometimes, from a practical clinical perspective, sometimes these constraints are not really needed.
I think we should probably stop here because I think there is a lecture after us. But Terry is around this afternoon, so if any of you have a few more questions to him, please ask him then. Thanks again, Terry.